Hi everyone. In this video, we will learn monitoring and control of foam in a fermentation. Foam sensing and control. Foaming is a serious problem in most microbial fermentations. Now, why foaming occurs in a fermentation? What is the cause of foaming? It may be due to a medium component or some factor produced by the microorganism. The most common cause of foaming is due to proteins present in the medium, that is proteinaceous rich medium such as corn steep liquor, pharma media, peanut meal, soybean meal, yeast extract or meat extract. These proteins may denature at the air broth interface and form a skin or a film which does not rupture easily, and this creates foaming. Now, foaming can cause further removal of cells from the medium. The microbial cells which are growing in the broth, those cells are removed from the medium and they come onto the surface. This leads to autolysis and the further release of the microbial cell proteins which comes out from the cells after lysis will increase the stability of the foam. Now, if uncontrolled, this foaming or foam may have serious problems, physical and biological. To name a few, this may cause reduction in the working volume of the fermenter, invalid process data due to interference at sensing electrodes, so incorrect monitoring and control. Cells get deposited at the upper part of the fermenter. The air filter exits become wet. All these things lead finally to loss of the product or fermentation. So foaming is not good and it has to be controlled. How would you control this foaming? Common practice is to add an anti-foam during a fermentation when the culture starts foaming above a certain predetermined level. Now, what are antifoams? Antifoams are surface active agents. They reduce the surface tension in the foam and they destabilize these protein films by forming hydrophobic bridges between the two surfaces, displacing the absorbed protein and by rapid spreading onto the surface of the film. Commonly used antifoams are some of these alcohols, esters, and fatty acids and derivatives, particularly glycerides such as cottonseed oil, linseed oil, sunflower oil. Now, these anti-foam additions will depend on the process and the economic considerations. Let's see a automated foam sensing and control unit. This is the fermenter. This is the broth in the fermenter. You can see an agitator here with the impeller blades rotating. Now, this is the foam sensing and control system, monitoring and control system. Now, this system consists of a probe. You can see this is the probe. Now, what is this probe? This probe is actually inserted through the top plate of the fermenter. So this is the top plate of the fermenter, and through this probe is inserted into the fermenter. What is this probe? This is actually a stainless steel rod, and this stainless steel rod is insulated except at the tip of the probe. Okay, so this probe, what you can see is a stainless steel rod inserted to the top plate of the fermenter. It is insulated except at the tip, and it is set at a defined level above the broth surface. So you can see here, this probe is set above the broth surface. This is not touching the broth directly. This is not coming into contact with the broth directly, and it is above the broth. It's not immersed into the broth. So when the foaming occurs, obviously the foam will rise, and the foam will rise and it touches the tip of this probe. Now the tip of the probe, remember, is not insulated. So when the foam rises and touches the probe tip, a current is passed through this circuit of the probe. So as soon as the foam rises and touches this probe tip, a current is passed into this circuit. You can see the detector here, and with the foam acting as an electrolyte and the vessel, fermenter vessel, acting as an earth. Now you can see here that this detector system and the circuit is connected to a pipeline here, and in this pipeline there is a valve or a pump, and this pipeline is connected to another vessel, and this vessel is filled with the anti-foam. So there is a vessel which is filled with the anti-foam, sterile anti-foam, and this vessel is ready. This vessel, antifoam vessel or reservoir is connected to this pipeline, obviously because this antifoam will flow through this pipeline. You can't open the fermenter and add antifoam, and there are valves or pump in this pipeline, and this circuit is connected to that. So when the foam rises and touches this probe tip and the current is generated, this current activates this pump or valve or operates this pump or valve, so that automatically this pump or valve is open and the antifoam through this pipeline is released into this fermenter for few seconds. Now you would say that there is probe, we understood, that is the main component. There's detector that will detect the current and that will activate this pump so that the antifoam flows through this pipeline for a few seconds. Why is this timer incorporated into this system? It has a very important role to do. This process timers are routinely included into the circuit to ensure that the antifoam, which is released into this fermenter via this pipeline, has time to mix into the medium and break down the foam before this probe is programmed after a preset time interval to sense the foam level again. So this is an automated monitoring and control system. So this probe is monitoring the foam. When the foam will rise, it will touch the probe, and as soon as the foam touches the probe, a current is generated. This current will be detected by this detector, and automatically this pump or the valve will be opened. Antifoam will flow into this fermenter for a few seconds, and the timer will ensure that the antifoam has time to mix into the medium and break down the foam before the probe is set to sense the foam level again. So this was the foam sensing and control system, automated monitoring and control of foam. Now, is there any other alternative other than this to control the foam if we don't use this system? Alternatively, antifoam may be added slowly at a predetermined rate by a small pump so that foaming never occurs only and there is no need for a sensing system. So rather than using this automated system, what you can do is, you cannot, in this system, what I'll be teaching you now, you cannot wait for the foam to occur. They don't wait for the foaming to occur. The antifoam is added slowly at a predetermined rate via a small pump or valve into the fermenter continuously so that the foaming never occurs and there is no need for a sensing system. But these antifoams, you would say these antifoams are, there's an advantage to add an antifoam into the fermenter because otherwise foaming may lead to loss of fermentation. Yes, but the antifoams have their own disadvantages. Unfortunately, the concentrations of many antifoams that they use in industries reduce the oxygen transfer rate as much as 50%. Therefore, antifoam addition should be kept to an absolute minimum. That's why many may not go for this system. Okay, and whether they will use antifoam or no, and if they will use which antifoam they will use, whether oil or silicone or alcohol or ester, and whether they will use this automated monitoring and control system or whether they will add the antifoam continuously all the time, that depends from industry to industry and also on the process. The antifoam or a method used in one particular industry may not be used in another industry or may not be used for every process. So that depends from industry to industry and also on process to process. And is there any other way if we don't go for antifoam? Yes, there are mechanical foam breakers available, and these mechanical antifoam devices have been described, including disc, propellers, brushes or hollow cones, and they are attached to the agitator shaft or the impellers above the surface of the broth. And these mechanical foam breakers, the foam is broken down when it is thrown against the walls of the fermenter. There are other mechanical foam breakers. The other devices which have been manufactured include horizontal rotating shaft, centrifugal separators and jet sprayers. Unfortunately, these mechanical foam breakers sometimes have to be used in conjunction with antifoams. For example, vinegar fermentation. You can't use a chemical antifoam in vinegar fermentation, and vinegar fermentation is often foam. Why you can't use a chemical antifoam? Because it's not feasible, as the antifoams may decrease the aeration efficiency and vinegar fermentation is a highly aerobic process. And also vinegar is a food product, so chemical antifoams are not desirable. So a mechanical defoamer or a mechanical foam breaker is incorporated into the fermenters or vessels which are used for vinegar production or fermentation. So this was all about monitoring and control of foam.